In the well-fed state, a rise in plasma glucose elicits the release of insulin, which will then activate the insulin receptors in many tissues. Activation of the insulin receptor elicits a plethora of intracellular responses, including 3 anabolic responses, which are synthesis of glycogen from glucose, synthesis of triacylglycerol from fatty acids, and synthesis of proteins from amino acids. In general, insulin will promote the synthesis of glycogen from glucose, the synthesis of triacylglycerol from fatty acids, and the synthesis of proteins from amino acids. Importantly, insulin will inhibit glycogenolysis, lipolysis of stored triacylglycerol, as well as protein breakdown. Key insulin effects on the liver. Starting with the liver, insulin will stimulate glycogenesis or glycogen synthesis and glycolysis. These two glucose-consuming pathways will use up nearly 60% of all the glucose present in the bloodstream after a meal. Importantly, insulin through its receptor will also have 2 inhibitory effects in the liver. Through a cascade of intracellular events, insulin will inhibit glycogenolysis or glycogen breakdown and will inhibit de novo glucose synthesis or gluconeogenesis. Effects of insulin on glucose uptake by muscle and adipose tissue. By specifically increasing the number of glucose transporters, GLUT4, on the surface of these two tissues, muscle and adipose tissue will pick up glucose from the bloodstream in an insulin-dependent manner. These two tissues constitute a significant portion of body mass, and as such they can contribute to a significant amount of uptake of glucose from the bloodstream, reducing the amount of glucose in the bloodstream. This effect of insulin is specific for muscle and adipose tissue only. If you like this video, please subscribe. Thank you.